Alright, welcome to Unit 2 in Biochemistry 2. If you want good explanations for everything, again, go look at my other videos. They explain it in a lot more detail. This is just how I would look at the slides and what to really highlight. It's just another way of looking at it. Usually we're skipping through a lot of stuff in the beginning. Epinephrine, it's beta pathway. That's what we're going to use most often. We're also going to look at the alpha pathway later in this unit. I would know these second messengers really well, especially cyclic AMP. Always make note if it's a 7-TM receptor or not, and which ones use G proteins. So here's another version of your second messengers: diacylglycerol and inositol. These are lipid-based. For calcium, you might say it's a mineral or an inorganic molecule. You eventually will know all of these, but I wouldn't worry about it too much for now. You see it in a better form throughout the unit. So anything that looks like this, I'll be able to maybe associate the ones in red for maybe a quiz, but for a test, I don't think this will be as important. This is more just kind of a FYI sort of thing. So this is the visual way of showing the epinephrine beta receptor pathway. If this helps you, great. This is totally optional. I like to be able to draw this out. What I prefer, being more left-brained, is this slide right here. So again, watch the other video to show, learning the middle part first, and then what's actually happening in each step, and being able to explain all that. It's a good reaction to know. The adenylate cyclase reaction produces the cyclic AMP. So this is pKa. This is what it looks like. It's got the two regulatory units in the middle, bounded by two catalytic units on the outside. You need 4 cyclic AMP to fully activate it. They bind to the regulatory units, and then the whole thing dissociates. So this middle guy separates, and you have 2 catalytic units, which then become active, and they can go do whatever pKa does. pKa does many things. That's for later chapters. For now, just know the general pKa structure. Know that it dissociates when activated. This is another way of writing it out in letters, so make sure you can understand this and realize that this is saying the exact same thing. All of them have off switches, which I explain in the other video in a better visual, but this is what it's talking about, how to turn off the alpha subunit of the G protein. Or GTP ACE, GTP hydrolase are the same thing. This is the way to terminate the actual receptor by using receptor kinase and beta arrestin. To break down the cyclic AMP, this enzyme right here, abbreviated cyclic AMP PDE. So remember, if you turn off the second messenger, you turn off the main enzyme. You turn off pKa. So remember, glucagon, if you know epinephrine, you know glucagon. They do almost the exact same things in the body. They're used almost interchangeably. The main thing to know that we'll talk about later is glucagon is mainly acting on the liver. Epinephrine acts on most cells. cross out epinephrine and write glucagon there, but then everything else is exactly the same. Some good reading there. Again, quiz level right here, more FYI. Cyclic Gmp is produced by these enzymes here. It's used as another second messenger. Definitely know this here, your soluble nitric oxide activated guanylyl cyclase. These are membrane bound, here and here. And this one's cytosolic, floating around. Remember, it's synthesized by arginine, also known as the smallest signaling molecule. This is Viagra, the actual name for it. It looks very similar to the cyclic GMP, so know this pathway really well as well. There's always a question on this. This is explaining that pathway in text form. So this is epinephrine pathway 2, the alpha pathway that we use much less. This one is phospholipase C, so you have some lipid molecules here. Lipid, lipid, lipid. And so the alpha pathway results in PKC rather than PKA. Just showing some of those reactions. This is the visual version of the epinephrine alpha pathway, eventually resulting in PKC. I would know that the IP3 receptor is membrane bound on the endoplasmic reticulum. DAG is in the membrane, and that's where it binds with PKC. Also know PKC requires both calcium and DAG, diacylglycerol. It'll be semi or half activated with just this or this, but if it has both of them, it becomes fully activated, and this is what that's showing right here. FYI/quiz material. We'll review this slide of calcium. Don't worry about that. This is fun. Calcium comodulin, know that they work together a lot. Come up a little bit later. This one I've definitely seen on a quiz before, so it could come up again, but again, probably not going to be on a test to know all of these are related to calcium and comodulin. So insulin, a little more complicated. Tyrosine kinase receptor. So be able to tell this story really well. We'll see in the linear form, but I think it's good for insulin to be able to draw this whole thing out, tell the entire story, and autophosphorylation here, PIP2 to PIP3, and how those are on the membrane. And remember, this is PKB. So we've seen PKA, PKC, and PKB. PKB is insulin, and that's eventually going to activate PP1. After this unit, all you need to do is think insulin, PP1. So if you're only getting one message, make sure that's it. This is all of the reactions individually. So this is the linear version of it. Make sure you know this really well. Be able to go back and forth between this and this. So this takes a little while to digest. The main things to know are, again, it results in PP1. GLUT4 goes to the membrane to let in more glucose. PP1 does a whole bunch of things. So think of it, you have sugar at hand, so you can store some as glycogen. You're not going to break down any glycogen. You can store some as fat. You have extra energy to make cholesterol. And glycolysis increases in the liver. Unit 3 will be over. Glycolysis. That's using the sugar and converting it to pyruvate. So break this down, make it more simple. If this is I think the hardest way to read previous insulin pathway, so I don't worry about this too much personally, but if it helps you, great. This is all review from Biochemistry 1. Make sure you remember tyrosine. So again, you'll see a question from all these slides. If you've seen them twice, he thinks they're important. So this is what happens when fasting. Be able to trace this down. When you're fasting, you have not a lot of glucose, therefore you have glucagon, and then you go through that pathway resulting in pKa. If you eat, you're eventually going to secrete insulin, which goes through the entire pathway. PP1. PP1 has all of those responses plus GLUT4. Alright, that's the end of that unit. Good luck.